Hello again, everyone. Today we're going to be talking about vaccines. This is lecture number 22. Remember, you can look along in your book. I have figure numbers where I'm using figures from the book. I do have several figures, however, that are not from your textbook, but hopefully they're large enough that you can see them. So today we're going to be talking about different types of vaccines, how they're made and how they work. And then also adjuvants, which are an important component of vaccines for many different types of vaccines. But I thought I would start with, where does the word vaccine actually come from? And it originates from cows and cowpox. And vac or vache is actually the Latin word for cow. What was observed back several hundred years ago was that people who worked with cows, milkmaids that milked them, or farmers sometimes did not get smallpox. But they did get a weak version, and it was cowpox, because cows also get a version of smallpox. So they thought that maybe if they gave people cowpox, that would actually prevent them from getting smallpox. And they were correct. So they started infecting people on purpose with cowpox. They got a very mild infection and maybe a few marks on their skin, but then they never got actual smallpox, which could be deadly and also very deforming. So that was really our first vaccine. Now, back then, they did not know anything about immunology, so they didn't really understand what they were doing, but they did understand the idea of protection. So, how can we fight infectious diseases with vaccination? So, the basis of vaccines is that they try to mimic what we naturally acquire if we get infected. So if you were to get smallpox and you survived, you would never get smallpox again. And that happens with some other infections as well. But as you know, not all infections, such as malaria, that you can get repeatedly. Most of our current vaccines are based in sort of three forms, and they are attenuated pathogens, which means they're still alive but weak, a killed organism, or a recombinant protein where we're taking just a piece of a pathogen and using that to immunize people. Most effective vaccines generate antibodies because it's actually difficult to generate cytotoxic T lymphocyte responses, and we'll talk about why that is. And of course, the goal of a vaccine is to be long-lasting. Ideally, they would be lifelong, but most of our vaccines, we do need to get booster shots throughout our lives. Live attenuated vaccines were some of our first vaccines, and they're similar to a cowpox vaccine where it's actually a live organism. Currently, though, today, we can actually do genetic engineering or genetic manipulation to develop even more attenuated vaccines. The route of vaccination is important. Sometimes we're used to getting shots in our arm, but there are intranasal vaccines. There are oral vaccines. So we can take vaccines in a few different ways. Conjugate vaccines have been developed more recently, and they take advantage of our understanding about how T and B cells work together, and really how T cells can help B cells make an antibody response. And then we have peptide-based vaccines, where we're getting really down to the very small pieces of pathogens that can elicit immune responses. But we have to combine those with something to really stimulate a strong immune response, and that's an adjuvant. So adjuvants enhance immune responses, and our limitation is not in having things that stimulate an immune response, but in having things that are actually safe to use. We don't want to overstimulate our immune systems. And I'm also going to tell you today about some new forms of vaccines. Most of these are currently being tested, but very few, if any, are actually being used yet. They're still in the testing phase. And they take advantage of our ability to do a lot of genetic manipulations. So, effective vaccines need to induce long-lasting protection, but be safe and relatively inexpensive. So you have this table here that tells you about how you want them to be safe, you need them to actually give you protection, and you want to induce both neutralizing antibody and protective T cell responses. And again, getting cytotoxic T cells is not easy from a vaccine. And then, of course, there are practical considerations, and I won't be discussing too many of those today. So what we're going to do is go through a list of the different types of vaccines, how they work, how they're made, and then also advantages and disadvantages of each. So we'll start with an attenuated vaccine. It's a pathogen that's usually grown in vitro in order to try to weaken it so that it can still infect someone, but is unlikely to make them sick because it grows more slowly and doesn't induce as much inflammation. So I'd like you to think about what pathogens this might work best for. It's actually viruses. One, because we can grow viruses in cell culture in a laboratory. Viruses grow very fast, but also a lot of viruses have the ability to mutate, so we can isolate variants that might be weaker than the original. So that's sort of shown here, and this is how we generated some of our first vaccines in the 20th century, like vaccines for measles, for instance, or mumps, or polio. We grew the virus in cells that it could infect, but wasn't its first choice to infect. And so we kind of, instead of using human cells, use monkey cells. The virus then changes, so it adapts itself to the monkey cells, but that then means it's not quite as good as infecting the human cells. But that makes it attenuated, and then that makes it good to use for a vaccine. Now, more recently, we can do this in a more directed way by using genetic manipulation to actually modify or take out virulence factors so that the virus is still alive but is weaker and less likely to make you sick. And even more recently, there's testing for a malaria vaccine where specific genes are knocked out of the parasite, and then the parasite can still infect liver cells, but depending on which genes are knocked out, it grows a little bit and then stops. So you don't go on to get malaria as a disease, but since you're infected with the parasite, your immune system responds strongly and to lots of different antigens in the malaria parasite in the hopes that you can develop a memory response to those antigens that would protect you if you were ever challenged with a normal wild-type malaria parasite from a mosquito. So what are the advantages of an attenuated vaccine? Again, it's an actual infection, so it better mimics what happens in real life. And again, that's what we're trying to do with a vaccine. Mimic what would happen if you did get an infection and recovered and then had a strong memory response, about which you've learned in your previous lectures. Since it's an actual infection, you are going to get good antigen presentation and probably better memory than you're going to get from some of the other vaccines I'll tell you about. Importantly, you're likely to elicit both a cytotoxic T lymphocyte response, so a cytotoxic T cell response that can kill infected cells, as well as an antibody response. And all of that together is likely to give you good protection. Now, there are some disadvantages. What if you had an attenuated strain of a pathogen and it reverted back to its wild-type form? That could make an individual sick. If you are immunodeficient from a genetic disease that you've learned about, from an infectious disease, or because you're young or elderly, even an attenuated form of a pathogen could be too strong for your own immune system to handle. not everything can be attenuated. We've had the best success with viruses, and I showed you an example potentially of a parasite, but it's actually been more difficult to develop attenuated bacteria. And you can imagine, since these are all going to be live organisms, that they're expensive to maintain and keep alive and to store and to deliver to people. Now, along the bottom here, I have attenuated strains can be transmitted from one person to another. You could view that as an advantage or a disadvantage. In the 1970s, they found that in some cities, about 70% of children were vaccinated with the attenuated polio vaccine, which is an oral vaccine because polio is transmitted by a fecal oral route. And because that vaccine was alive, although it didn't make the children sick, they were still able to pass that weakened virus to other children. So those other children also got vaccinated, and that's a good thing. So then the vaccine rate was actually about 85%, even though only 70% of children got the vaccine from their doctors. Now, the bad side is, what if that vaccinated child transmitted the attenuated polio virus to someone who is immunodeficient? That could make that person sick. So that's why you can view this either as an advantage or a disadvantage. The next vaccines we'll talk about are killed vaccines. So this is a whole organism inactivated, usually by heat, and then injected into a person. So what pathogens does this work best for? Really, we don't have a lot of limitations here. You could do this with almost any pathogen that you can actually isolate and grow in a lab, and that would be the limitation. And we commonly use this for the flu vaccine, and that's how we get new flu vaccines every year. So what are the advantages and disadvantages of this type of vaccine? So you can use this in immunocompromised people because it's not alive. So it's not actually going to infect anyone. So then you have a lower overall risk of adverse events. This is relatively easy and cheap to produce, and you can also do it quickly. And again, using flu as an example, that's how we can develop new flu vaccines every year. Some disadvantages. Since it's not an actual infection, you're sort of tricking the immune response into trying to think that it is. But when you put those dead organisms in, they don't ever grow. So there's a limited time that your immune system can see these organisms presented to them by the innate immune system. So overall, you get a weaker response and a weaker memory response that's not long-lasting. You are extremely unlikely to get cytotoxic T cells, because if you remember back to antigen presentation and cytotoxic T cells, antigens that get presented on MHC class 1 need to be made within a cell. If you are giving pieces of a dead organism, it's never made within one of our own cells. So you only get antibody responses. If antibody responses are enough to be protective, then that's okay. But if you need cytotoxic T cells to try to fight off certain pathogens, intracellular pathogens, then a killed vaccine may not work as well. You often need multiple vaccinations to try to boost that response and keep alerting the immune system, hey, here's some more of this organism, even though it's dead, please make a response. There is still potentially a risk, though, that when you inactivate the pathogens, one One stray one remains alive, and then that one pathogen, remember, is a completely normal pathogen. It's not attenuated, so that could potentially cause an epidemic. This is very rare, but it has happened on 1 or 2 occasions. The next vaccine we'll talk about is a subunit vaccine. So now, instead of a whole killed organism, we're going to take very small pieces and usually individual proteins from an organism. They could be isolated from the organism. That's how we originally did it. Now it's usually done with recombinant DNA and taking the gene for that protein and making it in the laboratory. This works well for toxin or toxoid-based vaccines, for instance, the tetanus toxoid vaccine that we have. Those toxoids tend to be very immunogenic, meaning they stimulate the immune system strongly, and that's good in the context of a vaccine because you want to get a strong immune response. So what are the advantages and disadvantages of these types of vaccines? Obviously, they're very safe. There's no pathogen ever involved. It's just one protein. You're less likely to induce a bad immune response. For a lot of infections, sometimes immune responses to certain antigens actually cause you problems. Too much inflammation or looking too much like self. You can avoid that here with just one protein. If you need to, you could distinguish between whether somebody's vaccinated or infected, because if they're vaccinated, they're only going to have an immune response to that one protein, whereas if they were actually infected, they'll of course make immune responses to many proteins from whatever the pathogen is. Disadvantages. Sometimes the fact that it's so safe means that you don't actually generate a very strong immune response at all. You are also going to have a lot more cost involved here because to make the recombinant proteins and to purify them so you could use them in a vaccine is much more expensive. And again, you're going to have a limited immune response to just one antigen. If the parasite or the pathogen mutates, you will lose your protection if you only have immune responses to one small piece of that pathogen. You will need a lot of boosting, and then of course, in this case again, there's really no opportunity to generate a cytotoxic T-cell response. When you're just giving a protein, it's not made inside of our own cells, so can't get presented very well, if at all, on MHC class 1. The next vaccine we'll talk about is something called a conjugate vaccine. So it takes advantage of our understanding about how T and B cells work together, in that T cells can see antigen presented by B cells and then help those B cells to make more antibodies. So we'll talk about how this works again, and then advantages and disadvantages. So a conjugate vaccine is developed by linking pieces of antigens to which the T cells respond to those to which the B cells respond. And there's a picture of that shown here. And B cells, remember, can make antibodies to non-protein antigens. And this shows a response here to a polysaccharide, a sugar. So, what do? We take the polysaccharide from a bacteria. That alone can work as a vaccine, in particular in adults. And it actually, a polysaccharide on its own, because it has many pieces, can cross-link antibodies on the surface of the B cell, cross-link those B cell receptors, and then actually you don't need as much T cell help. And that's what we learned about as a T-independent antigen. So we can use just the sugars in adults. But infants are very susceptible to infections that have these polysaccharides as antigens. And in fact, just the polysaccharide alone doesn't work as a vaccine in infants. And that's because infants don't have those marginal zone B cells that we learned about in our B cell lecture that actually respond to those T-independent antigens. They only have regular B cells that need T cell help. And you can't get T cell help to a polysaccharide antigen because it's not a protein. And remember, MHC molecules can only present pieces of proteins. So how can we conjugate a protein to a sugar in order to create a vaccine that will also work in infants? And that's what's shown here. You have the sugar connected to a protein. The B cell will internalize that, and it can still digest the protein portion, present that on MHC class II, and that will get shown to a T cell, and then the T cell can still provide help to this T-dependent B cell. What are the advantages? Again, very similar to the subunit vaccine we just discussed. They're, of course, safe. There's no infection here. Again, very limited number of antigens, unlikely to develop an immune response to something you don't want that could cause problems. You can still distinguish between an antibody response and an actual infection. But the big advantage is this vaccine will actually work in very young children, whereas a regular polysaccharide non-protein vaccine cannot be used in children. Disadvantages, again, are similar to the subunit vaccines. This can be, however, even more expensive to produce than a regular subunit vaccine because not only do you have to make a recombinant protein, you have to link together two different pieces, a sugar, a polysaccharide, as well as a protein together. Again, the rest of the disadvantages are similar to the subunits that we already discussed. And in particular, again, you will not get cytotoxic T lymphocyte responses. However, with the conjugate vaccine to these types of bacteria, they're often extracellular, so that's not really as big of a problem because cytotoxic T lymphocytes wouldn't be helpful if you have an extracellular organism as an infection. We have one very successful conjugate vaccine that we've been using for about 20 years, and it's a meningococcal conjugate vaccine. We have it to one serogroup C. You can see here when it started to be used, the number of infections went way down. We don't have one yet to serogroup B, so those infection numbers still stay high. And young children and infants still get this infection quite often. Our next type of vaccination or immunization is called passive immunization. And that's actually taking immune sera, or really the antibodies, from one person and giving them to another. I'm going to ask you guys if you can think of what the most common form of this will be. And it's actually a mother to a fetus or a mother to a baby with breastfeeding. We know that those antibodies in a fetus or in an infant can be very protective. It's not practical to do passive immunization on a large scale, but somewhat recently, you may have heard that this was used to treat some of the people who had Ebola. They took immune sera from prior patients that had survived and gave that sera to patients who are currently infected. So on a small scale, this can be very protective, but it's not ever practical to use on a large scale. Next, we're going to talk about some new technologies, new types of vaccines that most of these are in testing, sometimes late testing and clinical trials, but very few are actually on the market and currently used. And they're DNA vaccines, recombinant multivalent vaccines. We're We're going to talk about what an epitope vaccine would be, and then some viral vector vaccines. And I won't tell you about parasites, but the same idea applies that we talk about for making a recombinant virus. You can also do that for some parasites, such as Toxoplasma.